Assalamualaikum and hello my student. Let's continue our online class on chapter 11 for the third time. Okay, based on information in this slide, typical characteristics of the leaf are broad, flat and thin. The broad, flat shape that belongs to the leaf increases the surface area related to the volume. Leaves are the primary organ belong to the plant to perform the process that we call photosynthesis. Special structure belong to the leaf that we call stomata, which can be observed on the both surface of the leaf, which consists of pore, and found on the leaf surface that regulate the exchange of gases, or that we can refer as the process of respiration, and could cause the amount of water loss via transpiration. Now, in this slide, we want to discuss about transpiration process that happen on plant. Transpiration often causes in wilting of the leaf that impact of transpiration on plant. But by the process known as transpiration cooling, or that we call evaporative cooling, as tree release water molecule into the atmosphere from the leaf via transpiration, the surrounding air is cooled as water goes from liquid to a vapor. Other impact of transpiration cooling are lower the temperature of the leaf, and then the second one, prevent the denaturation of various enzymes involved in photosynthesis and input for the metabolic processes happen in plant tissue. The rate of transpiration in plant is affected by several factors. For example, like temperature and movement, light, humidity, and so on. So this slide we wanted to discuss about stomata. So stomata, as I said in previous slide, is a structure that can be found upper or at lower surface of plant leaf. Okay, the main pathway of water loss through the leaf is normally considered to be via the stomata. Figure in this slide, this one, show the structure of stomata. This is stomata structure, the first one. Okay, when it open, it's opening. Okay, this is another stomata structure, but it's in terms of its hole or opening become closed. Stomata, based on this figure, consists of two gut cells. This is gut cell belong to this stomata structure. This is similar gut cell belong to this another stomata structure. These two gut cells, as I said just now, it will surrounding the hole or the aperture or the lumen belong to this stomata structure. The opening and closing of stomata is governed by increases or decreases of solute. What example of solute? Okay, that can cause opening and closing of stomata aperture or lumen. Such solute, which are potassium ion, or nitrate ion, or chlorine ion. This is an example of solute inside the gut cell that can cause whether the stomata lumen will be closed or will be open. Swelling of the gut cell results in stomata opening, since the entry of the ion, the solute that we discussed now, and water within them make them bigger, or gut cell become turgid, and those able to move away from each other, making the stomata aperture larger. In contrast, closing is an opposite mechanism, and result in shrinking of the gut cell, or gut cell become fuzzy when the influx ion occur. Some plants can develop different methods to reduce transpiration rate. For example, a group of plants that we call xerophytes are a group of plants that can tolerate dry conditions. For example, they can survive in desert due to the presence of number of adaptation to reduce the rate of transpiration. Example of structure adaptation are having stomata concentrated on the lower surface and located deeper in the leaf tissue. Next topic we want to discuss about chapter 11 entitled Transportation in Plant is uptake the transport organic substance. Learning outcome that can be derived from the topic, which are the first one, describe short distance transport of organic substance, define sugar source tissue and sugar sink tissue, and lastly, the third one, explain the long distance transport organic substance via flow vessel. Okay, now this is the next topic we want to discuss under chapter 11, talking about the transportation of organic nutrients inside the plant tissue using the flow vessel. In plant, in the process of photosynthesis, the main product is glucose molecule, but it is usually converted into the larger sucrose molecule in plant cell before transported into the plant vessel. The transportation of sucrose molecule from the leaf tissue to the rest of plant organ and from the store structure such as in the root is called as translocation process. Translocation in the flowerworm, the predominant nutrients transported are sugar in the form of sucrose molecule, amino acids and mineral, with sugar, which is sucrose molecule, being the most concentrated solute in the flowerworm cell. Flowerworm cell will flow in plant tissue from the leaf as a sugar source tissue to the nutrient needed part of the plant, such as the stem and root that we call as zinc tissue that needed that sugar molecule as its nutrient. In previous slide, I have mentioned about the soft and thin plant tissue. So what we mean by soft tissue? So soft tissues are the plant organs such as leaf that produces sugar molecule. Then the thin tissue meaning as plant organ that consume or store sugar molecule such as the root organ belong to that. Figure in this slide describe the mechanism or process by which sugar in the form of sucrose molecule are transported through the flow vessel from the zinc tissue to the zinc tissue. Okay, this mechanism of process that we call pressure flow. At the soft tissue, usually the leaf, sugar molecules are moved into the sieve element. So sieve element is referred as to the flowerworm cell. Through active transport, water follows the sugar molecule into the sieve element through osmosis, since water basically diffuses into the region of higher solute concentration. This water creates two-go pressure in the sieve element, which force the sugar and fluid down the flowerworm tube toward the zinc tissue. At the zinc tissue, the sugar are actively removed from the flowerworm and water follows osmotically so that the condition of the higher water pressure and low sugar pressure are created, driving the pressure flow process. Again, based on figure in this slide, 6 stages involved in the process that we call pressure flow. Stages along the pathway as follows. Number 1, this one. Water moves up the xylem through transpiration. Stage number 2. Sucrose from a source is loaded into the flowerworm tube through ATP, increasing the solute concentration in the sieve tube. The third stage, this one, the concentration gradient allows the water from the xylem to move basically through osmosis from the low solute concentration to the high. The fourth stage, which is due to the high hydrostatic pressure, the sap is pushed from the source to the zinc tissue. The fifth stages in pressure flow, the transport of sucrose molecule inside the flow vessel. The fifth one, sugar is unloaded by companion cell, lowering the solute concentration. And the last stage that involves in pressure flow to transport the sucrose molecule inside the flow vessel, which is the sixth one. This allows water to basically go back to the xylem vessel through osmosis. This is now low hydrostatic pressure in the zinc tissue, so the sap from the source flow to the zinc tissue. Now we want to discuss about the information related about this slide, entitled Movement from Sugar Source to Sugar Zinc Tissue. In the process called sieve element loading, sugar are transported into the sieve element and companion cell. So this kind of mechanism or process, another name belongs to this mechanism of process that we call as short distance transportation of sugar or sucrose molecule inside the plant tissue. Sugar become more concentrated in the sieve element and in companion cell than in mesophyll cell. Once in the sieve element or in companion cell, That complex sugar are transported away from the source tissue. Then this process that we call as export. Translation of sucrose molecule to the zinc tissue inside the plant is called as long distance transport. Figure in this slide shows sucrose manufactured inside the mesophyll cell, this one. And then it can travel via the simplest pathway, this one. Okay, this arrow. Okay, to the cell member. So at the beginning, mesophyll cell will manufacture or develop sucrose cell, and then later on it will transport. Okay, that sucrose molecule, whereby it will pass in between ED cell before it will unload it, that sucrose molecule inside the cell element. Okay, based on this. In some species of the plant, sucrose molecule that manufactured inside the mesophyll cell, within the cell that we call phloem, parenchyma cell, it will pass up into another pathway before transported to the cell element. So this kind of pathway that we call upper class pathway. So this happens to the some plant. Okay, in general, sucrose that manufactured inside the mesophyll cell only will use simplistic pathway to transport or translocate into the cell element, but some other plant, they will diverse in terms of its direction go to the cell element, whereby when it enter the phloem parenchyma cell, it will pass out into the apoplastic pathway before later on it will go inside the cell element. This is another alternative for the some plant in terms of transportation of sucrose molecule inside the plant tissue. In the form of ATP molecule and two protein molecules that embedded in membrane cell belong to the plant cell, called as proton pump protein molecule and co-transported protein molecule that help plant cell to transport hydrogen ion and sucrose molecule. This mechanism of process enable for the plant cell to accumulate sucrose molecule in cell and input inside the companion cell as well. Continue our discussion in movement from sugar sources to the sugar sinks, transportation of sugar molecule inside the plant tissue. In movement from sugar source to the sugar sink tissue, the higher sugar pressure drive movement of phloem cell by backflow, or that we call mass flow, by positive pressure from the source tissue to the sink tissue, where the sugar in the form of sucrose molecule are rapidly removed from the phloem at the sink tissue. This pressure flow mechanism can be found inside the angiosperm plant tissue and include in gymnosperm plant tissue as well. Experiment in this slide was conducted to test pressure flow hypothesis. This study was done by a group of scientists. They used aphid, so aphid is an insect, to measure set flow and sugar concentration along plant stem tissue. The procedure for the experiment, the first step, aphid uses stylate to tap into the phloem. Next step, aphid body cut from the stylate after stylate is inserted into the phloem. The third step in protocol for this study, phloem sap continues flowing through the stylate. Then analyze sap for solute or carbohydrate. Later on, plants are grown in radioactive carbon dioxide gases, which become incorporated into carbohydrate produced by the plant. The sixth step involved in this study, radioactive level carbon can be detected in the phloem sap. And the last step in this study that involved of stylate places at different parts of the plant can show rate of movement of phloem sap. Then later on, what the result that can be obtained based on this study. The closer the stylate was to the sugar source, the faster the sap flow, and the higher was its sugar concentration. The conclusion for this study is stated based on in this slide. The result of the search experiment support the pressure flow hypothesis.